Today, I'll take up B lymphocytes or B cells, their maturation, that is their formation, then their activation and differentiation. Today, we'll take up first B cell maturation process. As I had told you earlier, the B cell, the term was coined because of Bursa fabricius, from which the B cells were discovered in the case of birds. But in humans and mammals, it is found that the B cells mature in bone marrow. The B word stands for bone marrow also. So the origin of B cells takes place in the bone marrow from the stem cells. There is two lineage. The major two lineages are myelocytic lineage and lymphocytic lineage. The myelocytic lineage will give rise to all the cells of the leukocytes that are macrophage, dendritic cells, basophils, eosinophils, and neutrophils. That is myelocytic origin. The lymphocytic lineage will give rise to T and B lymphocytes, and they have specific markers on their membrane, and those markers are called as homing receptors. And the B lymphocyte that is supposed to be mature in the bone marrow will have a receptor on its surface called as CD45 receptor. CD45 receptor. That makes the lineage of the B cell to remain in bone marrow. And when we are going to talk about T cell lineage, we'll talk about that separately in future lectures. But for today, for the time being, you keep in mind those lymphocytic lineages of cells which have the receptor on their surface which is expressed having CD45, they will remain in bone marrow to undergo further maturation process to give rise to B lymphocytes. And once they remain in bone marrow after their origin, they are called as pro-B cells. Pro-B cells. Pro stands for progenitor B cells. The pro-B cells, which are remaining in the bone marrow microenvironment, okay, go through two different processes in their maturation. One, the antigen independent phase of maturation, then antigen dependent phase. Okay, so antigen independent phase of maturation will be having pro-B cells. Now these pro-B cells which remain in the microenvironment of the bone marrow, because of their residence there, they develop a receptor on their surface which is called as VLA4. VLA4. VLA4 stands for variable lymphocytic antigen. Variable lymphocytic antigen. The pro-B cell first expresses variable lymphocytic antigen 4, VLA4. Once it expresses that, it will try to establish the contact. That receptor will try to establish contact with a molecule that is expressed by bone marrow stromal cells. And these stromal cells look like spindle-shaped fibroblast-like cells. They are in plenty in the bone marrow. They are very, very important. And they are responsible for large number of different processes that take place in the bone marrow for the maturation and development of our immune cells. Now those bone marrow stromal cells will express a receptor on their surface which is called as VCAM1. VCAM1, that is VCAM, that is supposed to be variable cellular adhesion molecule. Variable cellular adhesion molecule 1, and that establishes the contact with VLA4 expressed by pro-B cell. Once this first contact takes place, of the pro-B cell with the stromal cell through VLA4 and VCAM1, there is a signal transduction in the cytoplasm of pro-B cell. And that signal transduction will result in expression of another gene that called as c-KIT gene. c-KIT. c-KIT gene is the major cytokine gene family. Large number of cytokine receptors, large number of cytokines are produced under this family of gene that is called as c-KIT gene. So that c-KIT gene expresses on the pro-B cell. Now pro-B cell has two receptors, one VLA4 and c-KIT. And c-KIT receptor, once it is expressed, which is also called as c-KIT ligand, okay, it will establish the contact with another receptor expressed by the bone marrow stromal cell. And that receptor is called as SCF. SCF stands for stem cell factor. The stem cell factor establishes the contact with c-KIT. Now, pro-B cell has two receptor contacts. One is VLA4 contact with VCAM1, c-KIT contact with SCF1. This is called as the receptor synapse. Here, two different types of cells, that is pro-B cell, has established the synapse with bone marrow stromal cell through the two receptor contacts. Once this synapse is established, the pro-B cell expresses the gene for a cytokine, because I already told you c-KIT is the major gene for expression of cytokine receptors. Now, the IL-7 receptor, which is expressed by the pro-B cell, will start expressing on its membrane, all right, and that will become receptive for the interleukin 7 secreted by the stromal cells. Okay, interleukin 7 is going to be secreted by the stromal cells. To receive that signal, the receptor will be expressed under the command of c-KIT gene. Once the IL-7 receptor is expressed on the pro-B cell, that cell now is called as pre-B cell. From pro, it becomes pre. Pre for precursor B cell. Pro for progenitor B cell. There is some change in the morphology. There is some change in the gene expression. Okay. Once the IL-7 receptor is expressed and it responds to the IL-7 secreted by the stromal cell, that pro-B cell, sorry, the pre-B cell loses the contact with the stromal cell. That synapse that was established with c-KIT with SCF and VLA4 with VCAM is lost, and that pre-B cell becomes free. And after it becomes free from the stromal cell under the command of IL-7, which is being secreted copiously by the stromal cell, the pre-B cell undergoes mitotic expansion, in the sense mitotic proliferation or mitosis. Large number of clones of pre-B cells will be produced. Large number of clones of pre-B cells will be produced. It will be in millions. Each time it will be in millions. So once these clones of pre-B cells are produced, because of the response to the IL-7, okay, in the nucleus of the pre-B cell, some genetic change will take place, because B cell is designated to produce the antibodies after its differentiation. So it has to now start getting ready to respond and start producing the antibodies. For that, first it has to establish the receptor on its surface, a receptor to receive the antigen, a receptor to recognize a foreign antigen, to in fact interact with the foreign antigen. It should express a receptor, and that receptor it is going to express on its membrane is called as membrane-bound IgM molecule in the pre-B cell clones here, which is labeled as immature B cells, which are derived from the pre-B cells. After the expression of the antibody gene, the first antibody gene that is going to be expressed always will be IgM, immunoglobulin M. And that immunoglobulin M gene that is expressed by the pre-B cell, that molecule becomes the receptor of the B cell. And that B cell receptor, which is in short form called as BCR, B cell receptor. Okay, to start with, first it will express the heavy chain of the receptor, then the light chain of the receptor will be lost. Before that, the B lymphocyte also expresses, the B cell, pre-B cell expresses two more receptors, that is Ig alpha and Ig beta. Okay, and these two receptors, Ig alpha and Ig beta, which are also called as CD79A, that is alpha, CD79B, that is Ig beta, in pairs they will be accompanying the IgM molecule. MIgM, M for membrane bound IgM. It is a monomer. It has a heavy chain, that is the Fc region, which is lodged on the membrane of the B cell, and it has the two FAB sites. It is a monomer, two FAB sites exposed for receiving the antigen. FAB for fragment of antigen binding. All right, and always one MIgM molecule is accompanied by two pairs of Ig alpha and Ig beta adjacent to each other. Why? I will talk about that when I'm going to talk about the details of B cell receptor. For the time being, you keep in mind this expression of the receptor takes place in a stepwise manner. First, two receptors that are going to be expressed will be Ig alpha and Ig beta. Okay, that is CD79A and CD79B. And then the IgM is going to be expressed. That MIgM accompanied by Ig alpha, Ig beta will help in that pre-B cell now to become an antigen receiving cell. All right. Before that, one more receptor is going to be expressed on the pre-B cell, that is IgD. Now, why these two IgM and IgD molecules are expressed? The reason being, these two genes are present as the first sequence of antibody genes in the chromosome, and they are very close to the variable region of the gene for the antibody. The variable region gene first chooses these two constant region genes of the IgM and IgD. Therefore, these two antibodies are always expressed first, and they become the receptors on the B lymphocyte. But the major receptor of the B lymphocyte is IgM, and IgD becomes subsidiary. Similarly, even though the Ig alpha and Ig beta is expressed first, they also become subsidiary receptors. They help in assisting MIgM. But I, I tell you that this sequence of diagram, which is shown to you here, I do not expect you to remember this. This is only for your understanding purpose. Please remember that this region, that formation of pro-B cells, then conversion of those pro-B cells to pre-B cells, and expression of the receptor up to that stage is antigen independent phase, which is shown on top here. It is mentioned as bone marrow. In the bone marrow, the clones of pre-B cells will be produced with the receptors that are already present on them because of the influence of IL-7. Now they are ready to leave the bone marrow. After they express the receptor, they are ready to leave the bone marrow. That is, in this condition, here immature B cells In this condition, with the full-fledged receptor expressed on their surface, that is MIGM, they are ready to leave the bone marrow. Once they leave the bone marrow, the immature B cells, they are also called as now naive B cells, NAIVE. NAIVE is called as naive or newly formed B cells. They are not yet committed to any antigen. They are just formed. Very, very premature B cells. They are yet to come in contact with the antigen. Now, these B lymphocytes, which are immature B cells, once they leave the bone marrow, they will have to come and lodge themselves in the lymph node. Remember, I had done this in the cells and organs of the immune system, and they arrive through these afferent channels of the lymph vessels. From the afferent channels, they arrive in the cortex area of the lymph node, and they aggregate there to form the primary lymphoid follicle. Okay, the primary lymphoid follicle in the cortex area. There they have to now wait for the foreign antigen contact. At the same time, you remember that the B lymphocytes have every chance of coming in contact with the self antigens. Yes, they have to know the type of self antigens that we have. They have to learn to tolerate the self antigens. If they don't learn to tolerate the self antigens, these B lymphocytes themselves can become the active B lymphocytes to cause autoimmune response, which does not happen. So once the B lymphocytes have come and lodged themselves in the primary lymphoid follicle of the lymph node, they are ready to now receive the antigen. The MIGM now has the two ways of contact with the antigen. Either it can have established contact with the self antigen, because there are self antigens in plenty in our body and they are circulating in our blood, circulating in the lymph, and there is no chance that B lymphocyte can escape the self antigen. Okay, they will come in contact with the self antigens in large numbers. As soon as they have established this receptor on their surface, that receptor will have to come in contact with the self antigen. Now, once they establish contact with the self antigen through MIGM receptor, if there is a high affinity interaction means the self antigens will form a very firm binding with MIGM. Those B lymphocytes have to undergo negative selection. The negative selection is a process which will result in removal of the B lymphocytes which react with the self antigens with high affinity. These B lymphocytes, when they establish the contact with the self antigens through MIGM receptor, they are eliminated from our body. They undergo apoptosis because the self antigen, moment it establishes the contact with the MIGM, that will trigger the B lymphocyte with high affinity interaction to undergo suicidal activity, that is called as apoptosis. And if the B lymphocyte does not show any interaction with the self antigen, if it does not show any interaction with the self antigen, it has the time duration of a week to survive, 1 week. Those B lymphocytes that establish contact with self antigen are by apoptosis, 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 which is called as programmed cell death. And the process of deletion, the clones of B lymphocytes, that is, let's say, these clones of B lymphocytes that originated from the pre-B cells, if they establish the contact with the self antigen, all those clones will be eliminated. And that elimination process may happen in the bone marrow itself if they have come in contact with the antigen in the bone marrow as soon as they formed into pre-B cells with antigenic receptors, or when they arrived in the lymph node through the afferent lymphatic vessels. In the lymph node, they came in contact with the self antigen in large numbers, and those affinity interactions, which are strong interactions, that will eliminate them. Then the remaining population of B cells that are there, if they have not established the contact with the self antigen, they will wait for the non-self or the antigen of foreign origin to establish contact with them. Okay, and that establishment of the contact with the non-self antigen by the B lymphocyte makes that B lymphocyte selected for development into a fully mature active B lymphocyte and then undergo activation. That B lymphocyte survives. Within a week's time, if the B lymphocyte has not come in contact with any foreign antigen, then also that B lymphocyte has to undergo death. It gets frustrated that there is no antigen contact, there is no work to do, nothing it can do, and it will remain there in the primary follicle for a week. After that, it dies. Okay, but within that time, if it has come in contact with the non-self antigen, it will undergo positive selection. So there are two types of selection of the B lymphocyte. Okay, in the antigen independent phase, there is no antigen contact. The B lymphocytes undergo maturation. They become pre-B cells. Once they become pre-B cells, they have to undergo two types of selection. One is negative selection, and that negative selection is elimination of the B lymphocytes that are self-reactive. When I say self-reactive means those B lymphocytes established contact with the self antigen. If this self-reactive B lymphocytes are not eliminated, the person will suffer from autoimmune response. If they are allowed to undergo activation with the self-reactive antigen, they will cause autoimmune response. Because of that, they are eliminated. And that process of elimination is apoptosis, and it is also called as clonal deletion. Okay, then the second process of selection will be positive selection, which also happens either in the bone marrow or in the lymph nodes when the B lymphocyte, which has established the receptor on its surface, comes in contact with the non-self antigen. And that non-self antigen will trigger that B lymphocyte to undergo activation. But remember, first thing, the non-self antigen is not known to the B lymphocyte. Therefore, the receptor that is developed on the B lymphocyte, that is MIGM receptor, whenever it interacts with the non-self antigen, and that interaction will never be high affinity interaction. It will be low affinity interaction. That low affinity interaction with the non-self antigen is sufficient for that B lymphocyte to undergo activation. Alright, and once it undergoes activation, it has to undergo mitotic proliferation. And once it undergoes mitotic proliferation, in that series of mitotic division, it will do the permutation and combination of the genes of the antibody to see to it that it will establish or produce a receptor, MIGM receptor, on its surface with high affinity for that particular foreign antigen. Okay, and the first contact of the B lymphocyte, which happens in this cortex area, with the non-self antigen will be always low affinity interaction. Okay, and after 15 days of the first contact with the non-self antigen, the new population of the B lymphocytes that arrive with the proliferation due to the activation of the non-self antigen, that new population of the B lymphocyte will come with a receptor on their surface with high affinity interaction. Okay, high affinity interaction, and that high affinity interaction will be the one which boosts the immune response, and large quantity of the antibody production takes place, and that will go on from 15 days to 30 days, and the antibody production peaks means it goes to a very high titer, and that response will be sufficient to get rid of the antigen from the body. Okay, so in the lymph node, we had mentioned that the primary lymphoid follicle will become a germinal center and becomes a secondary lymphoid follicle where the proliferation of the cells will take place, right? And that proliferated cells migrate to the central area, that is paracortex area. So we had labeled the peripheral lymph node area as the light zone area, light zone. The paracortex area is called as dark zone. Dark zone is supposed to be rich with T and B lymphocytes, which are already activated with the foreign antigen, and they have undergone proliferation. There is a rich population of T and B lymphocytes in the paracortex area, which is called as dark zone. Then comes the medulla area. Medulla area is sparsely populated by T lymphocytes, whereas it is the B lymphocytes that are more in number in the medulla area, but they are not B lymphocytes anymore. They are called as plasma cells. Okay, in the medulla area, the B lymphocytes have undergone differentiation to become the plasma cells, and these plasma cells are responsible for the antibody production. So, copious amount of antibodies produced in the lymph node in the medulla area, and that antibody is drained to through the lymph node through the efferent lymphatic vessel, and it goes to the different parts of the body. Okay, so the B lymphocyte, the chain of events or the events that take place in the lymph node can be just summarized like this. But I don't expect you to remember this diagram. This diagram is not essential. For you, you are expected to remember this diagram. It is very important that where all the B cell activation takes place, where the proliferation takes place, and then the aggregation of large population of congregated cells will be there in the paracortex area, and then the antibody producing cells, plasma cells, will be there in the medulla. That's all you need to know. Other than that, this is all only for the understanding purpose. Remember, we have come across a dendritic cell called as follicular dendritic cell. And we had mentioned that follicular dendritic cells are supposed to be responsible for the presentation of antigens on their surface through the antigen antibody complex on their surface to the B lymphocytes. Now, let me call those B lymphocytes in two different terminology. One is called as centroblast, centroblast. Blast for proliferation. Blast for proliferation. Centrocytes, they do not divide. Please remember, those B lymphocytes which are highly proliferative, that is in the secondary germinal center, secondary follicle, and in the paracortex area, are highly proliferative B lymphocytes. They are called as centroblasts. Okay, and those cells which arrive here in the primary follicle area, they are called as centrocytes. They do not divide. Then once again, from paracortex area, when the cells which are now conditioned and programmed to develop the antibody against that particular antigen, when they migrate from this area to the medulla area, initially they are also called as centrocytes. Then they become the plasma cells. Please remember, the difference between centroblasts and centrocytes is that centrocytes do not divide, centroblasts divide. The B cell that is committed to the non-self antigen is activated, it proliferates. Now, in that proliferative process, the receptors that are put on the B cell surface could have high affinity receptors. I told you when it develops the activation with foreign antigen, it should develop high affinity receptor. Only those B lymphocytes are going to be selected for the foreign antigen. If the high affinity receptor is there for the self antigen, those B lymphocytes are going to be eliminated by the negative selection process. Okay, and the positive selection is the process for selection of B lymphocytes for non-self antigen, and they will develop high affinity receptors only after several cycles of cell proliferation. Now, if those B lymphocytes that are activated against the non-self antigen, if they do not develop high affinity receptor and they still keep the low affinity receptors, they are also going to be eliminated by apoptosis. Once they have come in contact with the non-self antigen, they cannot remain with the receptor of low affinity on their surface. They need to put in high affinity receptor by permutation combination of that antibody gene. Apoptosis is the only answer for that elimination. Those cells will be useless. Same way, the centrocytes that come in contact with the antigen that is being exposed on the surface of follicular dendritic cells, with antigen antibody complex on them, those centrocytes, if they do not show high affinity interaction with that antigen, then they are also supposed to undergo apoptosis. All this process of apoptosis is taking place in this light zone area of the paracortex. Light zone area of the paracortex. Okay, and if the high affinity interaction has taken place with the foreign antigen, these cells, high affinity interacting cells, have to move to the area of the dark zone in the paracortex region, and in the paracortex region they must now establish contact with the T lymphocytes to acquire long-term immunity. The B lymphocytes which have come in contact with the foreign antigen should establish a synapse with the T lymphocytes in the paracortex area. And if they don't establish the contact with the T lymphocytes, they will remain only as T independent B lymphocyte to develop the antibody, to produce the antibody, and that T independent B lymphocyte development process is called as TI antigen, T independent antigen B cell activation. Okay, the T independent B cell activation and T dependent B cell activation. Two different processes can take place depending on the type of antigen. Okay, and all that will take place in the lymph node area. Once the B lymphocyte has committed to the non-self antigen, it has to now come in contact with the T lymphocyte. If it comes in contact with the T lymphocyte, you have a long-term, lifelong immunity. That is T dependent, TD for T dependent antigen. And that establishment of the contact with the T lymphocyte through synapse of the receptors will make that B lymphocyte a important B lymphocyte that will have a long-term memory, and that T lymphocyte which established the contact with that B lymphocyte also will develop immune response against that antigen that is presented by that B lymphocyte on the class 2 MHC. Therefore, these two cells, the T helper cell, which came in first contact with the B lymphocyte, which is presenting the foreign antigen, will develop immunity against that antigen, and that is committed to that antigen, and it will become T memory cell. And that B lymphocyte which had presented that antigen to the Th cell on its class 2 MHC will become the B memory cell. Both these T and B memory cells, because of the TD antigen, TD for thymus dependent or T cell dependent, okay, T cell dependent antigen, will give acquired immunity and acquired immune memory for all the individuals lifelong. And these two cells will be stored in the bone marrow later, okay, bone marrow later. But there is also possibility of the B lymphocyte that is activated by a foreign antigen can undergo T independent stimulation, okay, T independent stimulation, proliferation, activation, okay. So that T independent antigens are classified into two types: TI1, that is T independent antigen 1, and T independent antigen 2. So what are these two different antigens? One, Ti antigen 1 is supposed to be lipopolysaccharides of the bacterial cell wall, LPS, lipopolysaccharide of the bacterial cell wall. Okay, and the lipopolysaccharides are a special category of the organic molecules that are there on the bacterial cell wall. They are capable of, see, two MIGM molecules are cross-linked by that lipopolysaccharide. Two MIGM molecules are cross-linked by that lipopolysaccharide, which is huge in molecular weight. So because of that, those MIGM molecules get stimulated in contact with the lipopolysaccharide and activate that B lymphocyte or undergo proliferation. Okay, and the TI2 antigen, TI2, TI2, T independent antigen 2. It is another special category of organic compounds of the bacterial origin or viral origin, and they are generally glycoproteins or complex proteins, glycoproteins, complex proteins, and the flagellins of the bacteria. Glycoproteins, complex proteins, and flagellins of the bacteria. Okay, and these molecules will establish that the type 2 Ti antigens establish contact with the MIGM in this manner, but this is not Ti antigen. It is shown here for TD, but the linking will be like this. Two adjacent molecules of the MIGM are cross-linked by one antigen. Whereas here, the entire antigen molecule is cross-linking two MIGM molecule, establishing the contact with the all FAB sites of the MIGM. Here, there is no need of establishing the contact with all FAB sites. Any two FAB sites of the adjacent molecule can be locked by the antigen, and that is TI2 antigen. This is the type of interaction takes place of the type 2 Ti antigen on the B lymphocyte. In both these processes of the Ti antigen, T independent antigen, the B lymphocyte that undergoes activation will produce only one type of antibody, that is one type of antibody, that is immunoglobulin M, and they will never become B memory cells. Please remember, Ti antigen, which establishes the contact, that is being foreign, contact with the B lymphocytes, succeeds in activating B lymphocytes and succeeds in making those B lymphocytes to differentiate to produce the antibody, but the antibody produced will be of only one class, that is IgM class. And these B lymphocytes will not give long-term immune memory because they have not come in contact with the Th cells. They did not establish synapse with the Th cells. So Ti antigen does not give long-term immune memory, and that acquired immune response is only immediate immune response, acquired immune response, and not long lasting. It will be sufficient to help the individual to overcome the infection. And once the individual overcomes the infection because of the IgM molecule, the immunity will subside and there will be no immune memory. Second time, the same antigen will enter, and when the same antigen enters, the same process will begin, and same process will result in production of Ti antigen stimulated B lymphocytes. Once again, the immune response will be there only for temporary. No immune memory. And these cells are not going to last long. Once the antigen load goes down, and once the antigen has gone disappeared from your body, those B lymphocytes will die. They are not stored as B memory cells. Please remember, T independent activation of the B lymphocytes is not going to give immune memory, and it is not long lasting. Only one type of antibody is going to be produced, that is IgM. And always the first antibody going to be produced is IgM. For long-term immune memory, always T-dependent B cell activation should take place, and that will be the establishment of the B cell contact with the T cell, and that happens in the lymph node. Thank you very much.